This is the second half of Chapter 12. We'll be talking about smooth muscle and smooth muscle contractions. So we can define smooth muscle by either its location, how it contracts, or how it communicates with the neighboring adjoining cells. As far as where do we find smooth muscles and its locations, smooth muscle is generally found in our vascular system, so our blood vessels. Gastrointestinal, the urinary, respiratory, reproductive, and even in the ocular, like when we were talking about our capillary muscle associated with the lens. So really all of our organ systems are associated with smooth muscle. Now, as far as contraction pattern, we have two types of contractile patterns. We have phasic smooth muscles and tonic smooth muscles, and I'll show you an example of that in just on the next slide. And finally, how they communicate, with two classes of smooth muscle. We have what we call single unit smooth muscle. Sometimes we refer to these as the visceral smooth muscles, in which they act as a single unit. And then we have these multi-unit smooth muscles, in which they're able to contract individual cells. Looking at the types of contractions of our smooth muscles, I stated that we have two types, phasic and tonic. The difference between these is how much tension is retained in that muscle when it's at rest. So in a phasic smooth muscle, at rest, when it's usually relaxed. So there's not an innervation going on that's maintaining tension in the muscle. Examples of this would be like in our esophagus or in our intestine, in which generally that tissue is relaxed. And then when we have an autonomic innervation of that muscle, it then builds tension in its contraction, and it's utilized for, in the case of the esophagus, peristaltic movement of food, a bolus into the stomach. Tonic, on the other hand, maintains tension throughout most of its innervation. So it's constantly being innervated, hence the name tonic smooth muscle. And so since it's always being innervated, there's also some amount of contraction that occurs. So you can see in this image, right on the phasic, we don't have any contraction force until we have innervation. Here, there's always innervation occurring, and then generally at some point that innervation will stop and you'll have relaxation, and then we'll go back. So our sphincters are a great example of tonic smooth muscle, in which we maintain some amount of tension and rigor in that muscle to do its function. Another great example would be in our blood vessels, right? Your blood vessels never truly fully relax, right? There's always some contraction occurring in that muscle itself. So that's the difference between phasic and tonic smooth muscles. Single unit versus multi-unit, it can be described by how that smooth muscle contracts. So in a single unit smooth muscle cell, what we'll see is that we have the varicosities that release the neurotransmitters, and then we have gap junctions between all of our smooth muscle cells, to where if we have contraction of one, it's going to allow those intracellular calcium concentrations to be shared throughout all of the cells. So we'll have contraction in each of these smooth muscle cells. So it contracts as a single unit. Innervation of one has a result in all of them. In the case of a multi-unit, however, you'll notice we don't have those gap junctions. And so we'll have individual cells contract for themselves depending on whether there is binding of neurotransmitters released from the varicosities. So when we look at our the muscles in the eye, right? This is a multi-unit, and this allows for a lot of fine tension control, because it kind of almost like recruiting additional motor units. You can think of each one of these muscle cells kind of like a little motor unit, if we're using that analogous compound in our skeletal muscle. Now, the rest of this chapter really kind of looks at differences between our smooth muscle versus our skeletal muscle, and how those are different from both a anatomical perspective as well as their contraction, how it's generated itself. And one of the most striking things that we notice about smooth muscle is that it is much more variable in how it is able to build its tension and maintain tension than either skeletal or cardiac. So we can see, skeletal muscle, it twitches very quickly. We build tension very fast, but then it's lost pretty quickly. Smooth muscle, though, takes a long time to contract. It also takes a long time to relax. From an anatomical standpoint, all our skeletal muscles are striated, and the contractile myofibrils run in the same direction. But in smooth muscle, we can have layers that run in lots of different directions. And so that means that the contraction is also going to be more varied and different than that skeletal muscle, where it's just a shortening or lengthening of the muscle. Smooth muscle uses a lot less energy than skeletal muscle to contract. I talked about how it can extend that contraction for a longer period of time. And then if we look at the cells themselves, what we see, unlike, say, skeletal muscle that are very large, they have multiple nuclei. Here, our skeletal muscles are small. They're generally that spindle-shaped cell, and they only have a single nucleus. They're not arranged in a sarcomere format. They don't have that same skeletal muscle structure. They get initiated by either electrical and or chemical signals, or sometimes both, where skeletal muscle is being initiated by an electrical stimulus, not a chemical ligand binding. It gets controlled by our autonomic nervous system versus the somatic motor nervous system for skeletal muscle. And the last part kind of gets to its contraction. The calcium that initiates contraction, just like in skeletal muscle, that calcium initiates contraction of our contractile elements. But calcium is brought in from the extracellular fluid, so from outside the cell, as well as from the sarcoplasmic reticulum inside the cell. And in skeletal muscle, it only comes from the sarcoplasmic reticulum. So the types of contractile elements that we find in a smooth muscle, it has a lot of actin, much more actin than in a skeletal muscle. However, that actin does not have troponin. We have a different system that ensures how the calcium binds and initiates contraction. We have less myosin, but the myosin filaments that we do have in smooth muscle are much, much longer. So a lot more actin, less myosin, and that myosin fiber has myosin heads along the entire length of it, whereas in skeletal muscle there's that middle region generally where we don't have heads. Lastly, we don't have T-tubules coming in from the cell membrane. We have something kind of similar called caveolae, but not quite as deeply innervated, mostly because we have all these gap junctions that bind the cells together in most of our single smooth muscles. So let's talk about our contraction and how it differs from that contraction we find in a skeletal muscle. So I mentioned that calcium can come from the outside. So intracellular calcium concentrations are going to increase when these channels get triggered to open. So we have these channels both in our sarcoplasmic reticulum and in the cell membrane itself, which brings calcium into the cell. When that calcium enters and we have that spark, it binds to a protein named chemodulin, or CAM for short. And what chemodulin does is, when it binds with calcium, it gets activated, and it triggers this cascade of additional enzymes and proteins. It activates what we call myosin light chain kinase. So it's inactive, but when we have the CAM calcium complex, that activates myosin light chain kinase. And myosin light chain kinase, the function that it serves is it phosphorylates the myosin head. It pulls that phosphate off. It cleaves it. Okay? So we can't split the ATP into ADP without myosin light chain kinase. We don't have myosin light chain kinase unless we have calcium bound to comodulin. So we need all of these in order for that power stroke to be activated. So this is different than in skeletal muscles where we don't have comodulin. Instead, we have troponin and this physical barrier that is preventing the phosphorylation of our myosin heads. Now, when we want to relax, how do we do that? Well, just like in the skeletal muscle, relaxation is going to be triggered by the removal of calcium. So we'll pump calcium back into the sarcoplasmic reticulum using a calcium pump. We also have a calcium pump on our cellular membranes that pump it into the ECF. And when we do that, and we're pumping calcium out this way using a primary calcium transporter, we then have to rely on restoring our membrane potential. And so we need to use a sodium calcium symporter to restore that. And as we start pumping that calcium out, again, we're going to have a dissociation with calcium with comodulin. When we have that, then we don't have any myosin light chain kinase activity, and instead we have a new enzyme called myosin phosphatase that's going to remove phosphate from our myosin chains and put it back into ATP. Okay. An interesting component of this contraction relaxation function of smooth muscle is that these smooth muscle cells can change their phosphatase activity. So if we go back, we just look, again, here's our phosphatase activity of how quickly myosin phosphatase is going to cleave the phosphate from our myosin heads and restore a inactive myosin head where we're not bound with our actin filament. And so this activity can be altered of how quickly we relax based off of calcium concentrations. And what we see is when calcium concentrations are low, like this would be a normal smooth muscle, right? So we kind of have this activity where here's the sweet spot in which we start to it desensitizes the myosin, and so myosin light chain phosphorylation and force of contraction gets greater, right? But what happens if that smooth muscle exists in a low calcium environment? If it's lower and we aren't able to, or the smooth muscle isn't able to adapt, we would never have this ability to form contraction. But what happens is that cell can alter that phosphatase activity where it becomes more, it sensitizes myosin and becomes more active at a lower calcium in that state to where we can still generate a full contraction, perhaps at a much lower calcium concentration. Likewise, if there's a lot of calcium present in the cell, we can desensitize the myosin to be less sensitive to myosin light chain kinase and myosin light chain phosphatase, and we can adjust in that manner. So this is an ability of the smooth muscle cell to become either desensitized, in this case, to intracellular calcium, or hypersensitized to calcium, depending on the needs of that cell. Lastly, smooth muscle cells are going to be regulated by both the sympathetic and parasympathetic neurons. So they're autonomic neurons that innervate our smooth muscle. That would lead to an electrical stimulation, right? However, we also mentioned that smooth muscle can be triggered by chemical, and those chemical signals would come from either hormones or paracrines that are being released from other perhaps neighboring smooth muscles. And so, an example of the paracrine would be histamine. So in your air pathways, right? When we're inhaling, if you're asthmatic, you may have an irritant that comes into your air pathway, and that irritant causes the cell to become irritated and start secreting histamine, which then histamine is a paracrine that may cause further contraction or constriction of that smooth muscle, making it harder to breathe. You would then have to take a bronchodilator, right? You take your asthma medication to dilate that smooth muscle. Nitric oxide is another example of like a paracrine that would relax blood vessels. There's numerous examples of hormones that can also bind to receptors that initiate contraction or relaxation of our smooth muscle. And this is different than what we see in our skeletal muscles. That's it. A lot of the last part of this chapter is knowing the difference between smooth muscle, cardiac muscle, which we haven't talked a lot about yet. It's in chapter 12 briefly. There's a great table at the end of chapter 12 that kind of puts skeletal muscle, smooth muscle, and cardiac muscle and talks about all the differences between them. But we'll talk a lot more about cardiac muscle next week when we move into the heart. So make sure you review that. Make sure you finish the Part 2 study guide and get that turned in as well.